Hi there. So my name is John Wilkinson. I'm an associate professor of chemistry and biochemistry at North Dakota State University. And I actually serve a couple of different roles on the NDACES project. And one role is that I'm a member of the Cellular Research Pillar. So this is one of the three predominant research groups involved in actually driving the project forward. Our job in the Cellular Pillar is to do a lot of the sort of testing of new materials to see which cells behave well and which cells don't, and how their behaviors change over time. So I have certainly a research responsibility as far as the NDACES project is concerned. The other role I play is that I'm actually co-lead of the Cellular Pillar. So I work closely with some of the other groups around, but part of my job is to help kind of make sure that the Cellular group is accomplishing the tasks in front of us and making sure that we're on pace to complete the research project in the time allotted. So that's kind of what I do. I'm like to be in the lab a lot. I like to get my hands dirty when it comes to actually doing experiments, but I also have a bit of an administrative responsibility as far as kind of helping to lead this project. As far as my actual research is concerned, so I have been interested for more than 15, going on 20 years now in mitochondrial biology. And so anyone who's taken biology going back as far as high school has been introduced to what mitochondria are. They're known as the powerhouses of the cell. That's the primary biological function that they've been determined to have over decades of research. And that's certainly true, and one of the things that I'm interested in. But it also turns out that mitochondria can actually be pretty important for triggering certain forms of cell death. So cell death sounds like a bad thing, and often it is, but it also turns out that cells die on a regular basis in the human body. The actual number of cells that die per minute is somewhat shocking. It's close to 1 million cells are dying in our body at pretty much any moment in time. And so my laboratory is interested in understanding how mitochondria balance this life-death decision that are often being faced by cells on a moment-to-moment basis. And more importantly, how that balance is altered in certain types of disease. So this is certainly an important feature for things like neurodegeneration, Alzheimer's disease, for example, as well as the primary focus of my group's interest, which has been understanding cancer development and progression. Cancer cells have to get energy in sort of creative ways, creative for the cancer, not so creative for the human being that's experiencing the cancer. And mitochondrial alterations are one of the ways in which that happens. And so that's been the interest of my research program for a number of years now, and I'm excited to be able to kind of apply some of that interest to the NDACES project. So as far as NDACES is concerned, so as I mentioned a moment ago, it's comprised of 3 predominant research pillars. There is a cellular pillar that I'm a part of, a materials pillar, which is tasked with coming up with new materials for the cell folks like us to test, and then a computational pillar, which takes some of the information that my group and my colleagues are generating and tries to sort of incorporate that into some mathematical models that might help explain cellular behavior. And so the primary responsibility that the cellular pillar has at the moment is to help use new materials that the materials group is designing, specialized and never used before chemical compounds that actually support the growth of cells in some unique and very specific ways. Our job is to test these new materials and see how do they work when we allow cells that are derived from either prostate cancer or breast cancer to grow upon these new materials. How do those cells behave? Do they work the way that we kind of expect them to? Do they tend to behave in ways that mimic the way an actual cancer cell might work in an actual human being? And if so, can we use that as a new way of testing cancer cell biology? Can we learn some things using these new materials that we weren't going to be able to learn previously? And so to give you a little bit more context about this, one of the questions that arises often is, why is this research important? And the answer goes back more than 70 years. And what I mean by that is researchers in cell biology, cancer biology settings have been using cultured human cell lines grown in plastic petri dishes for now going on 70 years. This actually goes back to the early 50s. And in that time, since the original experiments using cultured human cells were first demonstrated and developed that many years ago, the research community has learned a tremendous amount about how cells behave, how they are altered in disease states, and some of the ways in which drugs can actually be beneficial in preventing cells from behaving in ways that we don't want them to do in the context of, for example, a human being. At the other end of that sort of simplistic view of cells grown in culture, of course, are animal models. There are plenty of animal models that in many ways mimic human disease, which are also extremely beneficial in sort of developing science and being able to test drugs. And both of these, the simplest model, the cells on the right-hand side, and more complex animal models on the left-hand side, my right, your left, are extremely important, and they actually work together. But it turns out that neither one of those settings is exactly perfect, because while they have their advantages, they also have their disadvantages. And part of what this particular project, the NDASIS project, is trying to do is kind of find a middle ground between cultured cells grown in what I'll refer to as 2 dimensions, stuck to plastic petri dishes, and the more complex model that you might find in an animal, where you have multiple different kinds of cells that all interact with each other and communicate with each other in ways that influence their overall behavior. We call this 3-dimensional growth. And that's really one of the major focuses of the NDASIS project. Can we come up with new materials that let us actually allow cells to grow in these 3-dimensional conditions? Combine that with co-cultures, so more than one type of cell grown at the same time, and again, to see how they communicate with each other in ways that mimic in vivo, actually, in an animal behavior, but they're much easier to use. The animal models are powerful, but they can be very expensive. They take very, very long periods of time to complete. We're talking not just weeks to months, but we can talk about years, and in some cases, even decades to be successful. Not every institution can actually do animal research. There are certain requirements that have to be in place to be successful with an animal research program, and not every school, every university has that option. And so we're hoping that in testing these new materials that the materials pillar is developing, we're able to actually come up with some new ways to actually culture cells, and then do what with them? Well, learn again how they communicate with each other and how one cell can influence a decision or cell fate decision of a second cell might be obtained, as well as test again things like new therapies, drug delivery mechanisms, new types of chemicals that actually might serve as ways of changing cellular behavior that could be beneficial for a variety of reasons, up to and including human disease therapies that currently are not available. So it's a pretty important area of research. It has a lot of promise. I think that it's certainly a potential for transforming the ways in which cell biology is performed, and we're definitely excited to see this project progress. And so why might something like the NDASIS project be something worth considering for a student? Well, one of the things I think is great about this project is the scalability of the project. And what I mean by that is that there's no such thing as a simple experiment when it comes to how important any one experiment may be in deciding something new. But that doesn't mean that we can't actually engage this project at multiple different steps, depending on the skill set and experience of the researcher in question. So we can bring in people as young as high school level of experience in research and actually train them how to grow cells, how to develop these new materials, how to actually do some tests on the cells and see kind of how their behaviors are. Because there's lots of different things that we have to know about whether or not these new scaffolds are actually behaving the way that we'd like them to be behaving, which means that we can actually break this experiment, this overall project down into lots of different levels. And so it's actually pretty straightforward for us to engage students at various levels of experience and actually come up with ways to let the project grow with them as they get more experienced and more comfortable with the experiments moving forward. How do the students benefit from this in return? Well, long-term careers in biotechnology, biochemistry, medical sciences, even industrial type settings require a fairly strong research background. And I think the NDASIS project is well poised, well situated to provide that kind of a research background to students that can then have lots of different career options going forward. And so if I were actually trying to recruit a student to this project, what would I say to someone like that? If you're interested in learning how to grow cells in cutting edge, sort of state-of-the-art ways using materials that have never been used before, and maybe even coming up with ways to use these model systems as test beds for developing new drugs that may one day actually help improve human disease management and cures and that sort of thing, this might be the kind of project for you. So hopefully this has been an informative session, and thanks for your time.